Okay, the first in this series of short lectures, and you'll see it's very short lectures, just some revision notes really. Okay, so structure of carbohydrates. Carbohydrates are a major source of biological energy, we know, and they have the general molecular formula described there in brackets CH2O, N. N just means an indeterminate number. It's said to be the empirical formula for simple carbohydrates. So have a look at the formula for example, glucose, and check that you understand how this relates to the empirical formula. Now, when you get the complex carbohydrates like starches, this won't necessarily work. But have a look at glucose and maybe some other monosaccharides as well in your own time. Here's a general classification based on the number of monosaccharide residues in the molecule. So the little blobby thing there is a monosaccharide. Often glucose, could be fructose as well, of course, and a few other monosaccharide residues. So if it's just a single sugar, like glucose, we call it a monosaccharide. If it's two tied together, for example, sucrose or lactose, we call it a disaccharide. Then there's this interesting class called oligosaccharides, which are important for the metabolism of the bacteria in our guts. We can't metabolize them ourselves, but they are often important, such as for the metabolism of our passengers in our guts. Then the polysaccharides, really big molecules, containing hundreds, maybe thousands of residues, such as starch or cellulose. Okay, so the general classification. Say as an example, as I say, of glucose, it's a monosaccharide. It's defined as such because we can't break it down chemically to make a smaller sugar molecule. We can break it down to non-sugar molecules, however. As you'll discover in human metabolism, this molecule enters a process called glycolysis, where it's converted into 2,3 carbon fragments called pyruvates as part of our energy metabolism. The straight chain form I've drawn there is found in solid sugars, but normally in biology and in the lab we dissolve them in water and they transform to form a ring. Note the numbering system. In the ring form here, the carbon atoms and the ring of hydrogen attached are not shown for reasons of clarity. Again, have a look online, make sure you understand the full structure of glucose, because we will mention this occasionally a few times in a number of contexts. Okay, disaccharides, as I mentioned the examples a bit earlier, and the sucrose, which is a disaccharide of glucose and fructose, and lactose, which is a disaccharide of galactose and glucose. So disaccharides are defined as sugars which are composed of two monosaccharide residues joined by glycosidic bonds. So this is the glycosidic bond here and here. The most familiar one is sucrose. So when we hydrolyze these, in comparison to a monosaccharide, say we break it there, we end up with two sugars. So hence it's a disaccharide. Yeah, so there's the note the two different types of glycosidic bond in these two sugars, alpha 1,4 in sucrose and beta 1,4 in lactose. Check you understand why they're referred to as 1,4 bonds. We'll probably discuss this a bit later on when we talk about starch, because it's quite important. Some individuals and populations do not have the enzyme that breaks, that is to say hydrolyzes beta 1,4 bonds, which is where lactose intolerance arises from. Yes, as I mentioned, oligosaccharides aren't hydrolyzed by us, so they often said have a low caloric value, though they are hydrolyzed and used for energy sources by our gut bacteria. They're sometimes referred to as prebiotics. Gut bacteria, many of them are probiotic, and probiotics use prebiotics for energy. Not that many in nature. There's one of the common ones which is used in food, inulin. So you can see it's a chain of sugar molecules, basically, which we are not capable of digesting. But as I mentioned, our bacterial passengers can. Okay, polysaccharides, very large molecules, the largest found in nature. Think how big a tree is. Contain often uncountable glucose residues, not soluble in water, don't have a sweet taste. I should have mentioned monosaccharides and disaccharides have a sweet taste. Maybe so in the case of oligosaccharides, depends what they are. But polysaccharides are very large molecules, containing thousands of millions of glucose residues. Not soluble in water, not sweet tasting. The image there is of a transverse section of maple wood, which obviously produces maple syrup, which contains polysaccharides, which we can taste. Okay, so that's all for that.